Hello, this is our epigenetic approach to phantom limbs as discussed in the first couple chapters of the Ramachandran book, Phantoms in the Brain. My name is Emily Stonecipher. Normally the argument is on nature versus nurture, but today we're going to talk about epigenetics, or nature in combination with nurture. Epigenetics is when the gene expression changes, but the actual physical DNA does not change. Genes can be turned on or off when we're talking about epigenetics. On can also be called activation, and off can also be referred to as repression or gene silencing. There's a lot of different types of epigenetic modifications that can occur, but today we're going to talk about DNA methylation, histone acetylation, and non-coding RNA. DNA methylation is when a methyl group is added to the DNA. It's generally added onto the cytosine part of the backbone, which is paired with the guanine. The more methyl groups that add, the more like that gene is going to be repressed, and so more methyl groups equals more repression of those specific genes. Histone modification activates genes. It activates them by kind of loosening the chromatin, and that makes it easier for transcription factors to come by and use that gene to make more proteins. The third example is the non-coding RNA. It represses, it binds to the mRNA, and it ends up degrading the gene product. It self-propagates, and so it's been shown to have promising results in some cancer trials upcoming. Another good example of how epigenetics expresses itself, or how the environmental influences affect expression of genes, are bees. The worker and the queen bee have identical DNA. They're both females, and their only difference is nutrition. The worker and the queen have different functions, and they look different, but the queen is bathed in royal jelly when they're young, in their young larva stage. The royal jelly is an amino acid-rich protein, and it suppresses the gene DNMT3, and that ends up making all the difference in their life. Ramachandran mentioned a few different theories on why phantom pain exists, some of them being neuromas or neuron clusters or psychological factors. But his main theory seemed to be based on his patient Tom, that when the hand was lost, the sensory strip had been rerouted to the face, and when the face was stimulated, it also stimulated that same phantom hand, causing confusions and maybe pain in some patients. An epigenetic explanation for why some patients feel phantom pain and others don't is depicted in this figure on this slide. These twins share identical DNA, but through environmental effects on the acetylation patterns or the methylation patterns, these two patients had different experiences, one feeling no pain and one potentially feeling pain. The gene SCN9A codes for a voltage-gated sodium channel, and it's been found to be a pain marker or pain sensitivity marker. Mutations in this gene are found in people with extreme pain disorders or in people who do not feel any pain. There is an implied non-genetic risk to this gene, the mutations, using statistical evidence to subtract out genetic differences. Immediately following an injury, over 1,000 genes are activated in the dorsal root ganglion to cope. Inflammatory cytokine response is characteristic of injuries or autoimmune disorders. Glucocorticoids regulates and suppresses the cytokine response, and glucocorticoids are can be disrupted. Their expression can be disrupted from early life stressors like bad maternal care, diet, or bad grooming. Abused patients had disruption in their glucocorticoid production later on in life. Opioid regulators and central nervous system pain regulators are also have different expression changes right after an injury. Genes are switched on to help cope with the injury. Thank you for listening to my presentation. This is Emily Stonecipher, chapters 1 and 2 in Phantoms in the Brain by Ramachandran. Thank you very much.